Good morning. So I'll be providing a very brief overview of one of the most impactful applications of clinical epidemiology in medicine, and that's the grade approach for rating the strength of evidence and rating the strength of recommendations. So this will include a brief background of how grade started and how it was put into place. This will be followed by a brief description of the two main steps. One is the rating of quality of evidence, and following that, the process of going from evidence to recommendations. And finally, I will provide an example of going from evidence synthesis to finally issuing a guideline or a recommendation. So GRADE is an acronym. It stands for Grading of Recommendations, Assessment, Development, and Evaluation. As you may be aware, it's a transparent framework for developing and presenting summaries of the evidence and for providing a systematic approach for making clinical practice recommendations. It was in the year 2000 that the GRADE working group began to meet. So those were the people who put this process into place. It was comprised of international methodologists, so people who know about how to synthesize evidence, how to appraise research, and so on, and guideline developers. So there's a new breed of experts called guideline developers. The first publication that described this approach appeared in the British Medical Journal in 2004. Guidance for clinicians on applying and understanding GRADE appeared as a 6-part series in the BMJ in 2008. So this is where clinicians should go to if they want to know about what this is all about. And for methodologists, there were a series of articles starting in 2011 in the Journal of Clinical Epidemiology. So, and this series is actually continuing till date. So I think they're up to the 33rd or the 34th guideline document now. It's a process that's seen an overwhelming adoption and endorsement. More than 110 organizations have adopted the GRADE approach to rating the quality of the evidence. Included amongst these are the World Health Organization, the Cochrane Collaboration, leading American associations like the ACP Journal Club, or the ACP, the American College of Physicians, American Thoracic Society, the CDC, and the NHS. Also, leading hepatology associations like the ASLD and EASL follow and endorse these guidelines and the system. And also electronic textbooks like UpToDate and Dynamed that most of us carry around in our mobile phones. So it's a system that's going to be around a lot. We're going to be seeing this around a lot, which is one reason we should try to know about this. So what's all the fuss about this rating of the quality of evidence and so on? Why does it matter? This cartoon appeared in the BMJ in 2008, which sort of clarifies the issue of having to rate confidence in the evidence. So there's two weathermen talking in the Weather Bureau, and one says to the other, I figure there's a 40% chance of showers and a 10% chance we know what we're talking about. Which means that there's a difference between assessment of likelihood of an outcome and confidence in that assessment. And ultimately, the value of trying to determine the magnitude of an effect from an intervention, it depends on how confident we are about that assessment in the first place. So that's the basic issue that we're dealing with when we're talking about rating the quality of the evidence. With regard to medical curricula, it's become a core skill. So while earlier on critical appraisal skills were touted to be necessary to practice evidence-based medicine, that understanding is slowly changing. And even the world of epidemiologists, for whom critical appraisal is sort of a religion, they understand that clinicians and residents may not have the incentive or they may not have the time to actually inculcate the sophisticated methods of critical appraisal of original studies. However, understanding summaries, so understanding secondary sources of literature is a core skill in medical curriculum. So this is actually a very important skill to develop in medical training as well. And you can see what they wrote here in this article in the BMJ. Internal medicine residents could achieve evidence-based practice through secondary sources of evidence, such as trustworthy clinical practice guidelines and through feedback from the clinical mentors. That is, the ability to critically appraise independently was not a necessity for evidence-based practice. So what are we grading? Well, of course, first of all, interventions when we compare one management strategy to the other. Just a clarification here, what it is not about is individual studies. So we're not concerned with rating the quality of individual RCTs or observational studies. Rather, it is about complete bodies of evidence. So the complete summary of evidence on a particular clinical question is what we're concerned with. Ideally, this body of evidence should be summarized in the form of a systematic review and meta-analysis. And organizations like the World Health Organization that has endorsed the grade system, when they don't have a recent, up-to-date, and strong systematic review on a particular issue, then they commission it. So they ask some methodologists to actually develop and synthesize the evidence in the form of a systematic review. More recently, questions on prognosis and diagnosis have also been included, but what we'll be focusing on today primarily is interventions, therapies, so management. So what are we grading? There are two components. The first is the quality of the body of evidence, and there's a discrete categorization here. There are 4 categories: high, moderate, low, and very low. And the second thing is a simple binary system. That's the strength of the recommendation. So that's just either strong or weak. Now, when we talk about confidence or certainty in evidence, and all of these terms are used interchangeably, they're synonyms: confidence, certainty, quality, is referring to the same thing. One should think about it as a continuum. So going from no confidence at all to totally confident. So this totally confident would mean just do it. No confidence, you have to think about it, weigh in options and so on. And then there's a discrete categorization within this continuum. That's the 4 categories. And now this discrete categorization, of course, brings about arbitrariness, but the advantage of it being simple outweighs that. So how does this whole thing happen? Well, first of all, randomized controlled trials, they start out as high confidence. So an RCT would by default start at high confidence, and observational studies would start low. However, it's not necessary that an RCT will stay or will end up as high confidence. So there are a number of issues that will grade down the certainty or quality or confidence in the estimate from that body of evidence that comes from RCTs. Now, what are these issues that can grade down the evidence from RCTs or the summary of evidence from RCTs? There are primarily 5 issues, and I will talk just 1 or 2 minutes about each of these issues later on, but I'll tell you what these are right now. The first is the risk of bias. Second is inconsistency. The third is indirectness. The fourth is imprecision. And the fifth is publication bias. So any of these issues, if they happen to be there in the body of evidence, it will lead to a rating down of the evidence from high to moderate, low, and very low, and so on. Observational studies, on the other hand, although they begin as low confidence, but it's not necessary that they may stay as low confidence evidence. There are a number of things that can actually raise the confidence, and we can actually land up with high quality evidence from observational studies as well. Now, this happens most often when we have a very, very, very large effect from observational studies. For instance, there will never be a randomized control trial, and there never has been a randomized control trial on total hip replacement, because it's a large effect that happens very quickly. Similarly, for anaphylactic shock, epinephrine, for anaphylactic shock, there's no need for an RCT, because again, very large effect that happens very quickly. Dialysis for terminal renal failure, high quality evidence from observational studies. Similarly, furosemide for pulmonary. There are many examples where there's a very big effect, and so it precludes the necessity of a trial in the first place, and we get high quality evidence from just observational studies. And then finally, the third one is actually coming to the confidence. So you keep rating down RCTs because of these issues, or if they're observational studies, you will keep rating up, and we will land up with the final confidence, which I told you is 4 discrete categories ranging from very low to high. Any questions so far? Okay. So I told you RCTs start as high, observational studies start as low. There are 5 issues that can rate down the quality or certainty or confidence in that body of evidence that arises from RCTs. And these are those 5 issues. I won't cover, of course, there's no time to cover this in detail, but I will mention a little bit about each of these issues. But first, let me show you what this looks like when we have the product from the GRADE tool, right? So this is the evidence profile that we land up with when we apply the GRADE tool to a body of evidence. So this was a GRADE evidence profile for impact of surgical alternatives for pancreatic cancer. So they were comparing pylorus preserving versus standard Whipple pancreatic duodenectomy for pancreatic or periampullary cancer. You will see here that they have a rating for each outcome. So for each outcome, it's assessed what is the quality of the evidence. For example, 5-year mortality, in-hospital mortality, blood transfusions, biliary leaks, length of stay, and so on and so forth. And then you have these quality ratings for each of these outcomes. So you'll see here that there were 3 randomized control trials that reported 5-year mortality for this comparison. And so it started out as high, but then they rated down once for a risk of bias. And here in the footnote, you can see why they rated down. So it was unclear allocation concealment, patients blinded in only one study. So, all of these issues that I will talk about shortly. And so one rating down from high to moderate. So that's how it happens. Here, in hospital mortality, rated out twice. So once for risk of bias and once for imprecision, right? So that's how you land up with low quality evidence. So that's this part, the left part of the table. The right part of the table is the summary of findings table. So what this depicts, first of all, is the relative effect. So this is actually the result of pooling the results from those individual studies. And here you have the control rate. So the control rate can come from either the control group of the arms, control arm of the studies that you've included, or it could come from previous literature, whichever, that's a value judgment, which you think would be the best control event rate. And then over here you have your absolute effect. So how does this translate into actual events? So you can see your 5-year mortality, 20 less deaths, but it can range from 120 less to 80 more. Right? So this is how the results are depicted in an evidence profile. This is the product of applying the GRADE tool to that body of evidence. So talking about those 5 issues, first of all, the easiest one to understand is risk of bias. It's the most intuitive to know about. What this is referring to is basically the methodology of the included studies. And we're also familiar with all of those mechanisms in place to ensure the validity of results from RCTs, So there's allocation concealment, analysis should be on the intention to treat principle, blinding, completeness of follow-up. Those are the common ones. And more recently, there's also a focus on selective outcome reporting and stopping early for benefit. So, of course, in detail, we can talk about this in a session that's devoted to RCTs. What risk of bias is referring to is basically the methodological quality of the trial. So, of course, if it's not good, if we have unblinded studies, if we have unconcealed allocation, inadequate follow-up, not a robust analysis, then that would lead to rating down once. The second is inconsistency. What this depends on, again, this is an easy one to understand. Acceptability of results from a body of evidence depends on similarities across evaluations. So if you have a number of studies included in a particular systematic review, you would want that the result be consistent across all of those different studies. So similarity is the term for that is homogeneity, and dissimilarity you could say is heterogeneity. So you don't want to be clubbing results that look like apples and oranges. So that's what it's referring to basically, consistency. So very intuitive. The first way to detect inconsistency is by looking at the plot. So this is the standard forest plot that all of us are very used to looking at. This is the line of no effect, the line in the middle. It corresponds to a risk ratio of 1 or an odds ratio of 1 or risk difference of 0. And you'll see here that just by looking at the plot, you can make out inconsistency to quite an extent. So here you could see these results are fairly consistent, not so much here. So although this is a very intuitive process, what we're doing actually, again over here, you could see we're happy with the consistency of these results. And over here, you can see that these two and these two are actually not consistent. And it's not just about the direction of the effect. It's also that these confidence intervals do not overlap at all. Okay? So the criteria that we're using is, first of all, how close are the point estimates? So you'll see here that the point estimates are quite close. If you run a skewer through this, you'll be able to sort of overlap all of those confidence intervals. But you can't say that here because the point estimates are quite far away. So the first criteria is how close are the point estimates. And the second criteria is what I was just talking about with that skewer that's running through the confidence intervals. That's the overlap of the confidence intervals. So those are basically the two criteria that are used to assess consistency. Right, so the first method is just by eyeballing. That's visual inspection of the forest plot. And what you're looking at is those two things, consistency for overlap of confidence intervals and closeness of point estimates. But there's also statistics to help us out. And the statistic that helps us with consistency is the I-square statistic. It ranges from 0 to 100. So this is also a continuum, actually. But then again, there are discrete categories. More than 50% is where we would start to become concerned. And anything that's lower, so 0 to 30, 40% is maybe acceptable inconsistency or heterogeneity. So again, you can see here for this plot, you would probably come to an I-square of 0%. And for here, this is actually 89%. So those are basically the two things, eyeballing and I-square. Now, indirectness is a little less intuitive, but what it's referring to basically is whether you can apply the results of that body of evidence to your individual patients. So it's a matter of applicability. And what would be concerning is if the population's intervention comparators and outcome, they differ significantly between what is included in the trials and what would count as patients commonly. So say the body of evidence, if it includes older, sicker patients, you would probably be concerned applying that evidence to practice because the nature of the population is very different from what you're trying to apply to. Also intervention, so you could talk about low dose and high dose, or maybe different class of the same drug. Also comparators, so different standards of care. So standards of care may differ, and so that's another concern. Also outcomes, so while we may be concerned with long-term outcomes like cardiovascular outcomes and mortality, what the trials report is short-term outcomes, and you would have trouble extrapolating those results. For example, glucose control versus cardiovascular events. And one more type of indirectness is when you are interested in drug A versus drug B, but there's no direct comparison. And what we have is drug A versus placebo and drug B versus placebo. So that's a different kind of indirectness. So what we're interested is this, drug A versus drug B, but what we have is a comparison of each drug with placebo. And now there are things called network meta-analyses that help us out with this, but this is a form of indirectness as well. So any difference in population intervention comparison outcome or a difference in the actual comparison, that's what we're referring to when we're talking about indirectness. Okay. Now, probably the most vexing one is publication bias. It's probably the most troublesome to understand because it refers to evidence that's missing. Now, the problem is that studies with positive results are more likely to be published. So when the results of a study impact the possibility of publishing it, it's publication bias. But not only that, not only are they more likely to be published, they're more likely to be published in high impact journals. They're more likely to be published in English. They're more likely to be published faster. And so related to all of that, they're more likely to be cited as well. Now, how does this threaten the recommendations that arise from a body of evidence? And to try to get that across, I will just tell a story. Actually, there are two stories, and these are true stories. The first is actually a happy story. It's the story of thrombolytic therapy in myocardial infarction, so streptokinase. And this one is actually, touted as one of the great triumphs. Whenever somebody is asked to give evidence for evidence-based medicine, this is the example that comes to mind. This is what they like to cite as well. So, like any forest plot, this is the line in the middle is the line of no effect again. The horizontal lines are the confidence intervals, and this point in the middle is the point estimate. So all that is similar to a standard forest plot, but the difference here is that this is a cumulative meta-analysis, which means that all of these estimates are pooling all of the results of all of the trials that happened till that date. So they kept on pooling. So these authors, Antman et al. In 1992, they took a look back to see how did the story evolve with regard to trials on streptokinase in myocardial infarction, right? So all of these estimates are actually meta-analyses of all the trials till that year. So you will see here that by mid-1970s, with 10 trials and 2,500 patients, it started to become clear that, maybe streptokinase has some role in myocardial infarction. But, the confidence interval is still wide enough to warrant some uncertainty. We're not sure. By around 27 trials and 6,000 patients, it became pretty clear. So you have a sufficiently narrow confidence interval and quite a big effect as well, statistically significant. So it became clear by around it became clear in the 1980s. However, despite this rather convincing effect, despite the answer being in, there were a number of other trials that were carried out. And there were about, around 42,000 more people that were enrolled in randomized controlled trials, half of which did not receive the benefits of life-saving thrombolytic therapy. Now, why was this necessary? Well, because systematic summaries and meta-analysis did not come into existence until the late 1980s, right? So, had we had a meta-analysis earlier on, the answer would have been in right here in 1970s with 22 trials. However, it wasn't until a very big trial was held, the ISIS-2, that was held in the late 1980s, around 18,000 patients you can see here. That is when it came into practice. That is when people started recommending thrombolytic therapy. So we needed that big trial in order to translate that benefit into practice. Now, this is actually, as I said, it's touted as one of the examples of why we need, systematic summaries and meta-analysis, and why we need evidence-based medicine. However, the story of magnesium is very, very different. In 1991, there was a meta-analysis of 1,300 patients, and magnesium, and what they said about magnesium was it's an effective, safe, simple, and inexpensive therapy for myocardial infarction. However, when a big trial, the ISIS-4, 58,000 patients were included, it showed no benefit. So this was, said to be a blow to enthusiasm for both magnesium and meta-analysis, because in this case, the mega trial was at odds with what the meta-analysis recommended. And the story was very different from the thrombolytic therapy story. And these are the two classical examples when we talk about publication bias, because what went wrong with magnesium? Why did we make the mistake of saying there's an effect when there actually wasn't? Well, if you look at what happened, and this paper compared the magnesium story and the streptokinase story, you will see, first of all, that streptokinase required double the number of trials before a convincing effect was revealed. And what you'll also notice is that most of the trials in magnesium are smaller. So there were smaller trials. There were no big trials at all. Now, if you plot this, and this is how we actually detect, it's not a very good method, it has severe limitations, but this is how we detect publication bias. This is called a funnel plot. On the x-axis, you have some measure of the effect size, and on the y-axis, you have sample size. Now, ideally, if you draw a funnel here, it should be symmetrical. So these individual dots, these dots refer to individual studies. And if you draw a funnel here, the dots on either side should be symmetrical. And if it's not symmetrical, that's how you know that there's publication bias, because smaller studies, which occur towards the base of that funnel, they go missing. So in the streptokinase example, if you draw a funnel here, you'll see that it's symmetrical, and the estimate from the mega trial, the ISIS-2, is consistent with the meta-analysis of small trials as well. So there's no difference. And with regard to magnesium, small trials are missing. So this is the effect that publication bias has on the recommendations that arise from the evidence. Now, imprecision, I think I'll skip this because it requires more time. I'll just tell you this much, that imprecision refers to smaller sample sizes and small number of events as well. It leads to a very wide confidence interval, and so that reduces our confidence in the estimate. And how do we decide how much is too wide? Well, you look at the two ends of the confidence interval. If your decision changes, then that's an imprecise result. So when you go from evidence to recommendations, we have to weigh in the options, compare the benefits to the harms, and what we have to look at is, first of all, the quality of the evidence, and then the balance between the harms and the benefits. What is the significance of strong versus weak? First of all, there would be variability in patient preference. If there's a strong recommendation, all informed patients would, informed patients and clinicians would probably make the same choice of for or against an intervention. It impacts the interaction with patients. So if it's a strong recommendation, just inform, If it's weak, you ensure that it reflects values and preferences. And quality of care criterion. So if there's a strong recommendation, it forms a quality of care criterion. If it's weak, it doesn't. And then there's other things that go into consideration. And there are things like values and preferences, balance of effects, cost effectiveness, and so on. And finally, I will finish with this example of evidence synthesis from recommendations. Very early on in the pandemic, we synthesized the evidence for corticosteroids in COVID-19. There were no trials on COVID-19 at that time. So what we took into account is, again, indirect evidence from other coronaviruses, so MERS, SARS, and so on. What this evidence synthesis informed was a guideline. So this was the evidence-based guideline. It was published in May 2020. We started that evidence synthesis in March 2020. And the guideline panel made a weak recommendation in favor of corticosteroids in severe COVID-19 and ARDS. It was weak because it was rated down. One, of course, for indirectness, because there were no trials in COVID-19. And second, for risk of bias, because there were mostly observational studies. But what I wanted to show here from this slide was who was included in the guideline panel. And there are a number of different kinds of people: methodologists, end users, so critical care physicians, pharmacists, nurses, and also patient representatives. So these were actual patients, one severe and one mild COVID. There's frequently a patient partner or a caregiver. So a whole bunch of people, different kinds of people are there in the guideline panel who are explain the evidence, and they make values and preference judgments. Grade in ILBS, it can be inculcated into the residents' training through a workshop. Epidemiology department would be happy to synthesize evidence for important clinical questions. And we need a survey of values and preferences, because this is a highly context-specific issue. So in conclusion, GRADE provides a rating of quality of evidence and strength of recommendations. It provides explicit guidance that's transparent and informative. It's intrinsically not a simple process, but GRADE makes it as simple as possible. And it's widely becoming the worldwide standard, one may argue, deservedly or not. Thank you very much.